Military researchers documented in 1943 that a honey-based wound treatment eliminated infections on the battlefield. That research was filed away. 80 years later, peer-reviewed studies published in major microbiology journals confirm that Manuka honey destroys MRSA biofilms. These are the antibiotic-resistant bacterial colonies that prescription drugs cannot penetrate. 3 ingredients, $2, no prescription. A single course of antibiotic treatment for a resistant wound infection costs hundreds to thousands of dollars in the United States healthcare system. That assumes the pharmacy is still open. That assumes the antibiotic still works. The World Health Organization has designated antibiotic resistance one of the top 10 global public health threats. The compound you are about to learn costs roughly $2 to assemble from grocery store ingredients. It stores indefinitely on a shelf. By the end of this video, you will know the exact 3 ingredients. You will know the exact ratio. You will know the exact preparation method and both application protocols for a broad-spectrum antimicrobial powder that addresses antibiotic-resistant bacteria. And you will understand exactly why you were never taught this. The reason you have never heard of this has nothing to do with whether it works. Welcome to the channel. If you're new here, this is what we do. We recover buried practical knowledge, we verify it against the best available science, and we make it accessible to anyone willing to learn. This channel exists for people who refuse to be dependent on systems that can fail. Well, subscribing does not make you a fan. It makes you part of a knowledge network that is growing every single week. Every video we release is another piece of practical capability that you own permanently. No one can revoke it. No one can put it behind a paywall after you have learned it. It is yours. To understand why this compound matters, you need to understand what happened to it. 1,500 years before the Common Era, Egyptian physicians were documenting wound care protocols on papyrus scrolls. The Edwin Smith Papyrus and the Ebers Papyrus, 2 of the oldest medical documents ever recovered, describe the application of honey to open wounds. These were not folk remedies scrawled in the margins. These were systematic medical instructions used by trained practitioners in one of the most advanced civilizations on the planet. They observed that honey-treated wounds resisted putrefaction. They did not know the mechanism. They did not need to. They documented the result. Move forward 1,000 years and 1,000 miles northwest. Hippocrates, the Greek physician whose oath doctors still nominally swear today, documented the use of oregano for gastrointestinal ailments and respiratory infections. He also prescribed honey-based wound preparations. This was independent observation. The Greeks were not copying Egyptian medical papyri. They arrived at the same conclusions through separate clinical experience. Now move east. Ayurvedic medical practitioners in India were using honey in wound poultices and describing its preservation properties in texts dating back over 3,000 years. Their formulations were different. Their language was different. Their conclusions were identical. Honey prevented wounds from festering. The Romans documented similar practices. Pliny the Elder wrote about honey's medicinal applications. Roman military physicians carried honey as part of their field medical supplies. 4 civilizations, 4 independent traditions, 4 continents. The same conclusion repeated across millennia. Now jump to 1943. The world is at war. Sulfonamide drugs, the first mass-produced antibiotics, are being distributed to military field hospitals. They are revolutionary. They are also failing in certain wound types. Military medical researchers began documenting alternative wound care agents, including honey-based preparations applied directly to battlefield injuries. The results were documented. Infected wounds that had resisted sulfonamide treatment responded to honey application. Tissue granulation improved. Infection rates dropped. This was not fringe science. This was military medical research conducted under wartime urgency, when the only metric that mattered was whether soldiers survived. They documented it. They filed it. And then something else happened. Penicillin arrived at industrial scale. Pharmaceutical companies secured massive military contracts to produce it. The economic infrastructure of modern medicine began to crystallize around a patent-based, prescription-based, recurring revenue model. The honey research did not get classified in a vault with armed guards. Something more effective happened to it. It became irrelevant to the business model. It was simply never funded again. So if military medical researchers validated this in 1943, why did it disappear? The answer is not complicated. Follow the money. But before we do that, I want to tell you about the vault. What you're watching right now is the surface. Every video on this channel represents the publicly shareable layer of research that goes much deeper. The vault is the full archive. Field medicine protocols, water purification methods that work when municipal systems fail, food preservation techniques that do not require electricity, shelter construction from salvaged materials, perimeter security with zero budget. Every item in there has the same level of research backing as what you're learning right now. No filler, no fluff, no recycled blog posts. The link is in the description below. But you do not need the vault to use what I am about to teach you. Stay right here, because what comes next is the peer-reviewed science that confirms everything those ancient physicians observed. Now, let us talk about what modern science has confirmed about each of these three ingredients. And I want you to understand something. I'm not asking you to trust ancient wisdom on faith. I'm about to show you that 21st century microbiology laboratories have validated these mechanisms at the molecular level. Ingredient 1, Manuka honey, not regular honey from a grocery store bear bottle. Manuka honey sourced from the nectar of the Leptospermum scoparium plant, native to New Zealand and parts of Australia. What makes Manuka different is a compound called methylglyoxal, abbreviated MgO. Regular honey has trace antimicrobial properties primarily from hydrogen peroxide production. Manuka honey has that plus methylglyoxal at concentrations high enough to be medically significant. Here is what methylglyoxal does, and this is the part that should change how you think about infection treatment. Bacteria are not stupid. When antibiotics show up, many bacterial species respond by forming biofilms. A biofilm is essentially a communal fortress. The bacteria secrete a sticky polymer matrix and embed themselves inside it. Antibiotics hit the outer surface of that biofilm and cannot penetrate to the bacteria hiding within. This is not a theoretical problem. Biofilm-mediated infections account for a staggering percentage of chronic wound infections and implant-associated infections in hospitals right now. Imagine bacteria building a fortress wall around themselves. Conventional antibiotics hit that wall and bounce off. Methylglyoxal dissolves the wall. Research published through the University of Waikato in New Zealand, which has operated a dedicated honey research program for decades, has demonstrated that Manuka honey with MgO ratings of 250 and above disrupts established biofilms of Staphylococcus aureus, including methicillin-resistant Staphylococcus aureus, MRSA, the pathogen that terrifies hospital infection control departments worldwide. Peer-reviewed studies in journals including Frontiers in Microbiology and the International Journal of Antimicrobial Agents have confirmed this biofilm disruption activity. And here is what should stop you cold. The FDA has cleared Manuka honey-based wound dressings, long sold under the brand name MediHoney, for use in United States hospitals. Right now, today, burn units use them, chronic wound clinics use them. This is not alternative medicine. This is FDA-cleared wound care that is already in the supply chain of American hospitals. You were just never told about it. Ingredient 2, oil of oregano. Specifically, oregano oil standardized to a minimum 70% carvacrol content. Carvacrol and its related compound thymol are phenolic monoterpenoids. That is the technical classification. What they do, in plain language, is rupture bacterial cell membranes. Every bacterium, whether gram-positive or gram-negative, relies on the structural integrity of its cell membrane to survive. Carvacrol integrates into that membrane and destabilizes it. The membrane loses its selective permeability. The cell contents leak out. The bacterium dies. This mechanism is fundamentally different from how most antibiotics work, which is why bacteria that have developed resistance to conventional antibiotics have not developed resistance to carvacrol. The mechanism is too physically direct to evolve around easily. Studies published in the Journal of Applied Microbiology and the Journal of Food Protection have documented carvacrol's bactericidal activity against both MRSA and pathogenic Escherichia coli, including antibiotic-resistant strains. Gram-positive and gram-negative coverage, that is broad-spectrum activity from a compound extracted from a plant you can grow in a window box. Ingredient 3, activated charcoal, not briquettes from your grill. Pharmaceutical-grade activated charcoal, it's the same substance administered in emergency departments across the country for acute poisoning cases. Activated charcoal works through adsorption, and that distinction matters. Absorption means one substance soaks into another, like a sponge with water. Adsorption means one substance binds to the surface of another through chemical attraction. Activated charcoal has an internal surface area exceeding 3,000 square meters per gram. That is not a typo. A single gram of activated charcoal has a surface area greater than half a football field. That vast microscopic surface area electrochemically attracts and binds toxins, bacterial metabolites, and cellular debris. In the context of wound care, activated charcoal serves as the drawing agent. It pulls toxins and bacterial waste products away from the wound bed. It creates a cleaner microenvironment for the other two ingredients to operate in. Now here is where synergy matters. These three ingredients are not just stacked together randomly. They address infection through three independent complementary mechanisms operating simultaneously. The activated charcoal draws out toxins and bacterial metabolites, clearing the field. The Manuka honey dissolves the biofilm fortress that protects bacterial colonies from attack. The oregano oil's carvacrol ruptures the now exposed bacterial cell membranes and kills the organisms directly. Draw, dissolve, destroy. The ratio is 4 parts dehydrated Manuka honey, 1 part oregano oil powder, 2 parts activated charcoal, 4 to 1 to 2 by weight. That ratio was designed to balance the potent irritant properties of concentrated oregano oil against the soothing and moisture managing properties of the honey and charcoal. I will give you the exact preparation steps in a few minutes, but first, you need to understand why this is not in every first aid kit in America. So the science is published, the hospitals are already using one of these ingredients. The question you should be asking is why this compound is not standard issue in every emergency kit in the country, and the answer will make you angry. Let me walk you through the economics. Not conspiracy theory, just arithmetic. The global antibiotic market generates tens of billions of dollars annually. Companies including Pfizer, Johnson & Johnson, Merck, GlaxoSmithKline, and Novartis hold substantial positions in this market through patented antibiotic formulations. The business model is elegant from a shareholder perspective. A patented drug receives market exclusivity. A doctor writes a prescription. The patient pays. The insurance company pays. The cycle repeats with every new infection. Now, ask yourself a simple question. Can you patent honey? No. Can you patent oregano oil? No. Can you patent activated charcoal? No. Can you patent the combination? Not as a composition of matter, because all 3 ingredients are naturally occurring. Substances with documented prior use, stretching back thousands of years. If you cannot patent it, you cannot control the market for it. If you cannot control the market, you cannot generate recurring prescription revenue. If you cannot generate recurring revenue, you cannot justify the investment to your shareholders. And if no pharmaceutical company will fund the research does not get funded. It is not that the science was suppressed through some shadowy conspiracy. It is that the science was abandoned because no one could monetize the result. Consider this comparison. Neosporin, the most widely recognized over-the-counter antibiotic ointment in America, contains bacitracin, neomycin, and polymyxin B. It is effective against susceptible bacteria. It has 0 documented activity against MRSA. 0. The organisms most likely to infect a serious wound in a modern environment, the ones that have evolved resistance to conventional antibiotics, are invisible to the product sitting in nearly every medicine cabinet in the country. Meanwhile, Manuka honey with MGO 250+ has peer-reviewed published evidence of activity against those exact resistant organisms, and it costs less. In 1994, the Dietary Supplement Health and Education Act, known as DSHEA, established the regulatory framework that governs natural substances in the United States. Under this framework, honey, oregano oil, and charcoal are classified as foods or dietary supplements. They cannot legally make disease treatment claims without going through the full FDA drug approval process. That process costs hundreds of millions of dollars and requires a patent-protected product to justify the investment. Natural substances cannot receive meaningful patents, so the approval process is never initiated, so the treatment claims are never authorized. So your doctor never learns about it in medical school, so you never hear about it in a pharmacy. This is not a conspiracy. It is a business model. The information is published in peer-reviewed journals. It is not hidden. It is simply not profitable for anyone to tell you about it. If the information in this video is useful to you, the subscribe button is the only way to make sure the next one reaches you. We are building something here. A library of practical knowledge that does not depend on anyone's permission or anyone's profit margin. Every subscription strengthens the network. Every share extends the reach. Stay right here, because I owe you something important before I give you the instructions. I want to be honest with you about what this compound can and cannot do. Because unlike the industry we just discussed, I'm not going to overstate anything. What is proven beyond doubt? Each of the 3 individual ingredients has peer-reviewed antimicrobial evidence published in respected journals. Manuka honey is FDA cleared for wound care in United States hospitals right now. Activated charcoal is the standard of care in emergency medicine for acute poisoning. Oregano oils, carvacrol, and thymol have documented bactericidal activity against antibiotic-resistant organisms in laboratory studies. What is not proven? The specific synergistic efficacy of this exact 3-ingredient compound at the 4 to 1 to 2 ratio has not been validated in controlled clinical trials. The individual ingredients are validated independently. The combination is supported by mechanistic logic and anecdotal field use, but not by a randomized controlled trial. No such trial has been funded. You now understand exactly why, what you need to know about limitations. This compound is not a cure for every infection. That statement would be irresponsible, and I will not make it. Deep-seated tissue infections, sepsis, bloodstream infections, and viral, fungal, or parasitic infections are outside the demonstrated scope of these ingredients' antibacterial mechanisms. If you have access to professional medical care, use professional medical care. This compound is a field-expedient alternative for situations where professional care is unavailable, delayed, or where antibiotic-resistant organisms have rendered conventional treatment ineffective. Additional cautions. Oregano oil is a potent irritant at high concentrations. The 4:1:2 ratio dilutes it substantially, but sensitive individuals should perform a skin test on unbroken skin before applying to an open wound. Activated charcoal absorbs medications indiscriminately. If you're taking any oral medication, separate charcoal ingestion from medication by a minimum of 2 hours. Manuka honey contains sugars. Diabetic individuals should monitor blood glucose carefully if using this compound on large wound areas, as sugar absorption through wound beds is possible. This is not a miracle cure. It is a field-expedient antimicrobial preparation with individually validated ingredients and a sound mechanistic rationale. That is the honest assessment. And it is still more than most over-the-counter options can claim against resistant organisms. With all of that said clearly, here is exactly how to make it, how to store it, and how to use it. Materials. You need 3 things. First, Manuka honey with a verified MGO rating of 250 or higher. This is critical. Regular honey will not work. Clover honey will not work. You need Manuka honey with that MGO rating printed on the label. You can find this at health food stores, specialty grocery stores, or online. A jar sufficient for multiple batches will cost between $15 and $40, depending on size and source. Second, oil of oregano standardized to a minimum 70% carvacrol content. This must be stated on the label or certificate of analysis. Not oregano essential oil used for aromatherapy. Therapeutic grade oil of oregano with verified carvacrol content. Available at health food retailers and online. A small bottle costs between $10 and $20 and lasts for many batches. Third, pharmaceutical grade activated charcoal powder. Not capsules, although you can open capsules if powder is unavailable. Not briquettes. Not charcoal intended for aquarium filtration. Food grade or pharmaceutical grade activated charcoal powder. Available at health food stores, pharmacies, and online. A container costs between $8 and $15 and lasts for dozens of batches. Total cost per batch of this compound, approximately $2. The individual ingredient purchases will cost more up front, but each purchase yields many batches. Preparation, step by step. Step 1. Dehydrate the Manuka honey. Spread a thin layer of Manuka honey onto a silicone-lined dehydrator tray or a parchment-lined baking sheet. If using a dehydrator, set the temperature to no higher than 118 degrees Fahrenheit, which is 48 degrees Celsius. This temperature ceiling is critical. Higher temperatures degrade the methylglyoxal that makes Manuka honey medicinally active. If using an oven, set it to its lowest possible temperature and prop the door open slightly. Dehydrate until the honey is brittle and can be snapped into pieces. This typically takes 12 to 18 hours in a dehydrator, longer in an oven. You will know it is ready when it cracks like hard candy. Step 2. Grind the dehydrated honey into a fine powder using a mortar and pestle, a spice grinder, or a clean coffee grinder. The finer the powder, the better the final blend integrates. Step 3. Measure your ingredients by weight. 4 parts dehydrated Manuka honey powder, 1 part oregano oil, 2 parts activated charcoal powder. Use a small kitchen scale. For a practical starting batch, this could be 20 grams of honey powder, 5 grams of oregano oil, and 10 grams of activated charcoal. Step 4. Add the oregano oil to the charcoal powder first. Mix thoroughly. The charcoal will absorb the liquid oil and distribute it evenly through the blend. This is important. Adding the oil directly to the honey powder creates clumps. Charcoal first, then oil, then mix, then add honey powder. Step 5. Add the dehydrated honey powder to the charcoal oil mixture. Blend thoroughly until you have a uniform dark powder with a consistent color throughout. Step 6. Store in a dark glass jar with a tight-fitting lid or in a sealed mylar bag with an oxygen absorber. Keep in a cool, dark location. Stored properly, this powder remains viable indefinitely. The dehydrated honey is shelf-stable. The charcoal is inert until activated by moisture. The oregano oil's compounds are preserved by the charcoal matrix. Application. Topical wound care. Clean the wound with the cleanest water available. Pat the wound bed dry with a clean cloth. Apply a thick layer of the powder directly to the wound, covering the entire affected area. Bandage over the powder with a clean dressing. Reapply fresh powder every 12 to 24 hours, cleaning the wound gently before each reapplication. You should observe reduced redness, reduced swelling, and reduced wound odor within the first 2 to 3 applications. Healthy granulation tissue, which appears as a pink, pebbly surface, should become visible within days in a responding wound. Application, oral use. For gastrointestinal infections, suspected foodborne illness, or gut disturbance in a field scenario where medical care is not accessible, mix 1 teaspoon of the powder into 8 ounces of water. Drink 2 to 3 times daily. The taste is not pleasant. It is tolerable. Remember the 2-hour rule. Do not take this within 2 hours of any other medication, because the charcoal will absorb the medication and reduce its effectiveness. Perform a skin sensitivity test before applying to broken skin. Apply a small amount to the inside of your forearm on unbroken skin. Wait 1 hour. If no irritation, burning, or rash develops, proceed with wound application. You now know something that military medical researchers documented in 1943. Something that pharmaceutical economics buried for 80 years. Something that peer-reviewed science has confirmed in the 21st century. And something that 4 independent ancient civilizations discovered on their own, separated by thousands of miles and thousands of years, all arriving at the same conclusion. 3 ingredients, a ratio, a preparation method. Total cost, roughly $2. Available at grocery stores and health food retailers in any city in this country. Legal to purchase, legal to possess, legal to use, shelf-stable, indefinitely. You're now less dependent on a system that may not be there when you need it. That antibiotic supply chain is 3 days long in most American cities. 3 days from distribution center to pharmacy shelf. When that chain breaks, and history says it eventually will, the people who prepared will have options. The people who did not will have nothing. If one person you share this video with uses this compound when they need it, when the pharmacy is closed, when the antibiotics have stopped working, when the system they trusted has failed them, then this video did exactly what it was meant to do. Share it. Save it. Write the ratio down on paper and put it somewhere that does not require electricity to access. In the next video, we are covering another compound that was standard military issue for over a century before the commercial market found a way to replace it with something patentable and inferior. That one is already in the vault. The public version drops next week. This is knowledge that belongs to you. It always did. You just were not supposed to find it.